Once you have successfully run the pipeline, you can view the data plots and tables related to your datasets. To view these results, select any completed project from the Projects table, and notice a new table appears to the right. Verify that the files associated with this table correspond to the files that you're interested in, and click the View Results button. This takes you to the Results page, which initially shows you two different tables. The Project Information table illustrates the project information that you have defined, as well as the information inferred by the project during the pipeline process. For example, if you selected auto detection, the organism and strandedness are determined during the pipeline. The Status Details page illustrates the status of each of the jobs associated with your project. You want to verify that each of these are completed. However, it is possible that one of your analytics algorithms fails, but the rest succeed, so you can still view some results. Finally, the Download All Results button allows you to download all of the results on this page into an Excel spreadsheet so that you can further analyze it later. The first table on this page is the Raw Data Processing Report. This illustrates how well the pipeline algorithm aligned. First, you want to verify that the number of reads corresponds with what you believe each sample has. And then secondly, you want to make sure that the alignment rate is a high enough number so that you can be confident in the results displayed below. If you have many samples, this table will have multiple pages, which will illustrate each of these samples, reads, alignment rate, and Phred score. The Unsupervised Analysis report displays a hierarchical clustering algorithm, which was used to generate a heat map, as well as two different dendrograms on both the samples and the genes. In these dendrograms, you can filter based on clicking one of the branches and holding down. The same type of filtering can be applied to the genes. Highlighting a cell illustrates the value in more detail, as well as telling you which sample this cell belongs to. The sample names are also displayed at the bottom of the table, and at the top of the table you can see the different conditions that each of the samples belongs to. Clicking on the camera allows you to export this table into a PNG format for further review. The next step in unsupervised clustering is the PCA and TSNE data visualizations. This shows you the different clustering based on the different spaces defined by the leading to PCA components or the leading to TSNE components. If the sample names are overlapping each other, you can click the Hide Details icon to get a more detailed view of each point individually. The DIG report is broken into two sections. A summary section, which allows you to look at each of the different groups side by side, and a detailed version, which allows you to look at a single group in more detail. The summary table illustrates each of the conditions compared against the wild type condition. In this example, group 6 was defined as the wild type condition. Here, for each of these, we have a number of DEGs, and how many of these DEGs are downregulated and how many are upregulated. Using the Q-value cutoff, I can change what value of Q-value we allow to determine the number of DEGs. As this number increases, the number of DEGs should also increase. Finally, we can choose one of the conditions to study in more detail, and this illustrates the log of the fold change, the p-value, the q-value, and the gene function based on an NCBI table for each of the gene names associated with the organism that was determined with your experiment. Since, depending on the organism, there can be between 4,500 and 45,000 different genes, you can use this filtering text box to filter on one of the gene functions. For example, if we click acid, we can filter all the gene functions and gene names to only include the ones that contain acid in the function name. You can also filter on a specific gene name, and it will only show that gene. Just as in the first table, if there are multiple pages, you can scroll through using the page icon. The density distribution gives you a more summarized view of each of the different conditions by themselves. Here we've aggregated each of the expression levels for all samples in a given condition and plotted the density by fitting against a gamma distribution. Here we see the density for each of the different expression levels for each of the conditions. Finally, the Go report is another heat map and dendrogram example, except instead of filtering on genes, it's based on the NCBI Go terms, genetic ontology terms. Again, here we can filter on the different branches of the samples or of the Go terms themselves. And the far right column allows us to view the full Go term on mouse over. We can again export this table as a ping or reset everything using the refresh button. So those are the different analyses and results that are displayed in the MODA results page. Now you have the tools to analyze all your data at the click of a button. Thank you for using MODA and have fun taking control of your data.